For the sixth part of the series of lectures for adaptive immune response, so now we will talk about cytokines. So cytokines is like the cell's communication system. So this like their own language, consists of chemical signal. So this is how the cell talks or communicate between them. Cytokines are signaling molecules secreted by the immune cells. So almost all of the immune cells have will secrete some sort of cytokines. The main function is used by immune cells to regulate the intensity and also the duration of the immune response by either stimulating or starting or inhibiting or stop the response, either start or stopping the response, stimulating or inhibiting. So they have two ways. First, activation of cells. So if we observe here, either stimulate the activation of cells or also inhibit the activation of cells. Second, proliferation of cells. So this is the where the cell become clones multiply themselves into large numbers. Proliferation of cells. Differentiation of cells. For example, T cells differentiate into three types: T helper, T regulatory, T cytotoxic, and the memory cells. So that's the meaning of differentiation. Fourth, secretion of antibodies by plasma cells, and also secretion of cytokines by immune cells. They can be in the form of proteins, peptides, or glycoproteins, involved in autocrine, paracrine, and also endocrine signaling as immunomodulating agents. They produce in response to an immune stimulus. So immune stimulus is the things that can stimulate or start the immune response, such as invasion by pathogens, where infection. We can observe here the inducing stimulus, which will stimulate the cytokine producing cells to produce cytokine, and the cytokine produced will be received by the target cells. So every target cells have their own receptor. So the cytokine is very specific. So each cytokines have their own target. So means that the cell need to have their own specific receptor. Once the receptor meet the specific cytokines, this will start the signal for the activation of the cells, which lead to the target cell biological effects. For example, when the macrophages exposed to pathogens, they will release inflammatory cytokines. So inflammatory cytokines mean the cytokines that will enhance or start the inflammation. Second example, upon recognition of antigens by the APCs, T helper cell will release cytokines for the other T cells and B cells to activate and proliferate. So the actions of cytokine, first is by autocrine action, where self-stimulation. Autocrine activation is the cytokine binds to and stimulates the cell that produce it. So they produce the cytokines which can stimulate themselves or the same type of clone types. For example, T helper cell produce interleukin 1 and interleukin 2, So those interleukin can stimulate the other T helper cells. So this is the case of autocrine. Second is paracrine action. Paracrine activation is the cytokine binds to and stimulates cell nearby to the one that produce it. So this cell produce cytokines that stimulate nearby cells. So this is the paracrine action. And the third is endocrine action. In endocrine activation, the cytokines binds to and stimulates cell distant from the one that produce it. So this is where the released cytokines will go into the circulation, to the blood and the lymphatic fluid, and once they reach their destination, they will stimulate the distant cell. Here is the required reading for this lecture, so I will provide together in the near future, and these are the references for the lectures. Thank you very much.